friends, in this tutorial discussing about GABA receptor and drug acting on them. Okay, this is Mohd Asif. Let's get started. I will cover some important topics like introduction of GABA and GABA receptor, GABA A, GABA B, C, drug acting on GABA receptor, agonist, modulator, antagonist. If you're watching first time here, please subscribe my channel, hit the bell icon, drop a new video, and then you can get the notification of that video. Let's get started. When we talk about the introduction of GABA, GABA stands for gamma aminobutyric acid. It's the inhibitory neurotransmitter of mammalian brain. It's found basically CNS, central nervous systems. Okay, and other neurotransmitter, inhibitory neurotransmitter is glycine. Is also inhibitory neurotransmitter. It's found in spinal cord and brain stem. It's found in spinal cord and brain stem. You should keep in mind. It's a glycine is found in spinal cord and brain stems. Okay, and when we talk about the history of discovery of this background of this 19th centuries was known as the metabolite of plants and microorganisms. Early 20th century isolated amino acid in the brain of mouse through the petrography. Okay, and 1950 Robert and Franklin discovered GABA in humans' brains. And when we talk about the synthesis of this, and glutamate converted into GABA by the help of a specific type of enzyme that is called glutamic acid decarboxylase enzyme. This helps to convert it into GABA neurotransmitter. This is the diagram. You can this picture. This is the glutamine converted into the glutamate enzyme by the glutamine amine enzymes, and glutamate converted into the GABA by the help of glutamate acid decarboxylase enzyme, and GABA further converted into the GABA transaminase enzymes by the help of GABA transaminase enzyme that is called succinyl semialdehyde, and semialdehyde further converted into succinyl acid. Okay, and this is the picture of a nerve cell. You can see it in nerve terminal, presynaptic, synaptics, and cleft, and GABA and vesicles containing the GABA. GABA is a neurotransmitter, and GABA vesicles fuses the presynaptics and releases the GABA into the cleft regions, and GABA bound to the GABA receptor, and GABA receptor present on the postsynaptic cells. Okay. This is the terminal. When we talk about terminal, it reuptake into presynaptic terminal and surrounding glial cell and via reuptake transporters. And function of this inhibitory transmitter in the brain: cerebral, Purkinje neurons, cerebellar cortex, substantia nigra, plus pellitus, etc. 20% of CNS neurons, GABAergic, 30% of the synapses. A presynaptic inhibition within the spinal cords. Okay, presynaptic inhibition within the spinal cords. Okay, GABA receptor. When we talk about the GABA receptor basically is three types, major three types: GABA A, B, and C. A is for ligand-gated chloride ion channel, and GABA B is for GPCR. PCR stands for G protein coupled receptor, and GABA C it is for transmitter gated chloride ion channel. Okay, this is the GABA A receptor. When we talk about the GABA A receptor is the ionotropic receptor, also known as, and cis loop family. It's also glycine, nicotinic, and 5-HTH3 receptor. Distribution of postsynaptic membranes, other Leydig cells, and immune cells, liver, bone growth, and plates. It's distributed other parts also. Okay, don't confusing about this. And fast postsynaptic inhibitions is selectively permeable to chloride ion side of action side. When we talk about there are some drugs which comes under the GABA A receptors. Okay, comes in GABA A side of actions: benzodiazepines and barbiturate, ethanols, anesthetics, steroid, volatile anesthetics. This is the site of action which is very responsible for this. Okay, this is the structure of GABA. You can see this picture. This is the these basically three categories. Two two unit alpha, 2 unit is a beta, 1 unit is a gamma. Okay, this is the GABA receptor. You can see this picture. Okay, and benzodiazepines is basically bound to the alpha unit. Okay, structure pentameric transmembrane receptor, 5 subunit around the central pore, and between the and among these unit between middle portion of alpha, gamma, beta. Between the middle part of it, there is some specific type of pore. Okay, that's responsible for ion exchange. And two unit of alpha 1, 2 beta 1, and 1 unit gamma. Okay, and subunit measure most commons alpha 2, alpha 3, alpha 5. So this is the location of this and function, also composition, distribution, measure function of GABA alpha receptor, GABA A receptor. This is the GABA receptor, and I will cover in this paper. In this slide you can see this picture. This is the functions, and alpha 1, beta 2, gamma 2, widespread GABA neurons. Okay, and is the function of sedation, anti-convulsant activity. Anti-convulsant activity is nothing but it's like a seizure, epilepsy activity. Okay, and subunit composition alpha 2, beta 3, gamma 2, and 4 brain spinal cords found in, and anxiety, muscles relaxant, and alpha 2, beta 1, gamma 1, glial cells. And this is the composition of this and location of this. You can see this picture is different compositions. Okay, this is the very important slide. You can see this picture. This is the very important slides. Okay, and alpha 4, beta 2, delta, thalamus, tonics, inhibitions, and location of hippocampus, and subunit compositions alpha 5, beta 3, gamma 4, tonics. Okay, and alpha 6, beta 2, 3, gamma 2, cerebral granule cells, and gamma 3, theta, epsilons. Little information. It has not information because scientists are working on lots of research going on. When we talk about the mechanism of action, GABA A receptor, activation of GABA A receptor, endogenous GABA A, inositol, exogenous drugs. Okay, opening of central pore, and increase the influx of chloride ions through the pores, and hyperpolarization of neuronal membranes. Okay, and decrease the occurrence of mechanism of action potentials, and inhibition of neurotransmitter ultimately. Okay, and how decrease the action potential. Okay, and how inhibit process is going on through the GABA A receptor. You can this picture. This is the very interesting slide. When we talk about the allosteric modulation is nothing but it has the no activity its own, bind to side others than that of GABA. Allosteric side like positive allosteric modulation increase the action of neurotransmitter, that is called some special example of BZD. And negative allosteric modulation is decreases the action of neurotransmitter, the example of becquerelines. Okay, this is the special type of allosteric modulations, positive and negative also. And when we talk about the GABA B receptor, it's a special type of receptor which is very responsible, also known as a metabotropic receptor. It also known as G coupled receptor. Okay, and coupled inhibit the calcium and channels, activate the potassium channel, and when we talk about the distribution of GABA 2 receptor, GABA B receptor is a distribution central and autonomic division of PNS, peripheral nervous systems. Okay, when we talk about the some action of mechanism of action of this presynaptically autoreceptor, inhibit the voltage gated calcium channel that decrease the neurotransmitter release. Postsynaptic inhibitory activate the potassium channel and long-lasting hyperpolarization and slow positive synaptic inhibitions. Okay, you can see this slide. You can remember, keep in mind how is it going on process. When we talk about the structure of GABA B, this is the GABA B structure. Is the dimeric unit, okay, and two transmembrane domains, and GABA B1, GABA B2. This is the two types of GABA B1 and B2. Okay, this is the GABA and GABA B1's GABA B2 are, and G protein is found in the cytosolic domain of the GABA receptor. Okay. When we talk about the mechanism of action in details and how this process going proceed, this binding of GABA to the extracellular domain of a B1's region. B1 is allosteric changes in the B2 subunit coupled to G proteins, and venous pipe traps inhibit the adenyl cyclase, activate the potassium channel, decrease the calcium conductance, and decrease the neurotransmitter releases and action potentials. Overall, we can say that in simple words, because GABA B2 is a decrease the release of calcium ions, okay, and decrease the action potentials. And when we talk about the GABA C receptor, GABA C receptor is nothing but the least studied among the three major classes of GABA receptor. GABA is a more selective GABA C than GABA A, found in retina, spinal cord, superior follicles, pituitary pentamers of P subunit with chloride channel in the central GABA P. Okay, this is the picture of GABA A receptor, GABA C receptor. You can see and you can keep in mind how is going on. Okay, this is the barbiturate and other drugs. This is the comparison study of type, location, structure, mode of actions. GABA A, GABA C, GABA B. You should keep in mind easily for long times. And inotropics, metatropics, widespread postsynaptic and presynaptic postsynaptic and pentameric dimerics postsynaptic inhibition by increase the influx chloride influx, some decreases in presynaptic inhibition by the decreases the calcium entry, postsynaptic inhibition by increasing the permeability. Okay, endogenous agonist GABA. When this is the pharmacological effect, this is the pharmacological effect. When we talk about the pharmacological effect of the GABA A, it causes the sedation, hypnosis, anxiolysis, anticonvulsant, amnesia, muscle relaxation, aphasia, and GABA B muscle relaxation, epileptic nausea, suppression of drug craving, anti-nociceptions, cognitive impairment, inhibition of hormone release. Okay, this is the pharmacological effect of GABA A and GABA B comparison we study. Drug acting on GABA receptor. Okay, this is the agonist receptor side and modulator side. Muscimols, gobosomadols, and acamprosodate, isoguanine, progabide. Modulator side is the BZD and barbiturates, ethanol, steroid inhaled anesthesia, neuroactive steroid, topiramide, non-BZD, zolpidem, zolpidem. This type of drugs, antagonist drugs. Antagonist bicolins, because and modulator side amintofilazamine, inverse agonist, beta carbolines, channel blockers, picotoxin, and pentalitrazoles. This is the GABA sides agonist and antagonist, and barbiturate side depression excitant, and benzodiazepine sides agonist, depressant agonist, and inverse agonist, steroid side anesthetics excitant, picotoxin side convulsant, depressant, chloride side. Okay, is already discussing about it. Agonist, the benzodiazepines. It's benzodiazepines is a drug that causes the sedative hypnotics, and they uses the sedative, hypnotics, and anxiolytic anticonvulsant. Allosteric modulator of GABA A receptor bind to the accessory side benzodiazepine binding sites ligand acting at this side, and positive modulator, negative modulator. Positive modulator is a agonist. Negative modulator is nothing but inverse agonist. It's also antagonist. Okay, keep in mind. Mechanism of action: GABA facilitator action, GABA A receptor. You can see this picture. This is the benzodiazepine BZ binding site, benzodiazepine gamma subunit, and post-synaptic membrane. This is the post-synaptic membrane, and that chloride ion channel. Chloride is the cleft regions. This is the nothing but is already discussing about it. So barbiturate is a controlling. Barbiturate is also controlling, acting depressant, anticonvulsant, and anesthetics. There are some example of this: phenobarbiturate and pentobarbiturate, etc. And also GABA facilitator increase the duration of GABA carotid chloride channel openings. And high concentration anesthetics may be also GABA minematics, direct activating chloride channels. Very high concentration inhibitory effect, and less selective, less BZG also decrease the action of glutamic acid via binding to the AMP receptor, also exert non-synaptic membrane effect, multiple more pronounced central depressant effect, low margin of safety. When we talk about the general anesthetics also like GABA A receptor are major side of the actions. IV anesthetics, the example of this atomides, prophylines, barbiturate, and neuroactive steroid, volatile anesthetics, isoflurane, and inflammatory target, like that. Okay. Neurosteroid compound related to the steroid hormones like metabolites, progesterone, androgens, like acting like BZ activation of GABA A receptor, steroid receptor, delta subunit, anxiolytics, analgesics, anticonvulsants, sedative, hypnotics, effect general anesthesia, high doses. Also act on NMDA, AMP, glycine, serotonin, nicotinic acid, acetylcholine receptor. Alpha zolones. This is the synthetic neurosteroid development as an anesthetic agent, potential benefit in the RX of anxiety, epilepsy, etc. So, negative modulator of GABA A sulfated endogenous steroid, progenolone sulfate, DHEAS. Okay. When this is the alcohol modulator of GABA receptor, extrasynaptics, delta subunit containing GABA receptor, sedative, low concentration of ethanol, prolonged ethanol consumption, decrease the delta subunit expression in the brain. Acamprosate, GABA analogs to treat the alcohol depressant, partial GABA agonist, and weak the NMD receptor antagonist. In chronic alcoholism, down regulation of GABA A receptor and alcohol withdrawal, little effect on AP, unopposed sympathetic activations, okay, and increase the action of open chloride channel. Okay. Antagonist, bicolines, conversion alkaloids, competitive antagonist at GABA binding site, block the fast inhibitory synaptic potential. GABA zymes, synaptic GABA analogs action similar to that of the bicolines. Useful experiment tool, but have not therapeutic uses. Flomazazine synthetics BZD derivative. Imidazole specific antagonist, BZ binding site, modulator site, competitive antagonist, the binding of D and non-BZG hypnotics, and does not antagonize barbiturate. This is the special nothing but inverse agonist. Drug that bind to BZG side exert the opposite effect to that of the conventional BZ. The example of the beta carbonyls and diazepam binding inhibitors. Mechanism explained by two stated one which can bind GABA molecule and open chloride channel, and one which cannot be bind GABA. Okay. This is the picture. You can see the picture. BZ agonist like A, and then okay, you can keep in mind inverse mechanism B. Already discussing about direct channel blocker. This is the direct channel blocker. Some drugs have picrotoxin, direct channel blocker, plant product conversion, direct blocker, chloride channel of the GABA, non-therapeutics, pentyl antitrozole, experimental consent, penicillin blocks the channel by interacting the positive channel amino acid residue with the channel pore. Antagonist, therapeutics potential GABA A receptor. Agonist, insomnia, BZD, anxiety disorder, alpha zolams, epilepsy, chronic muscles spasm, spastic general anesthetics, pre-anesthetic medication, alcohol withdrawal. This type of agonist, which is very important for various therapeutics potential uses. Agonist potential uses of BZD, analgesic, adjuvant, psychiatric mania, schizophrenia, depression, alpha zolams, adenazolams, post-stroke. Okay. Antagonist, flumezanil, inverse agonist, BZD overdoses, reversal of BZ anesthetic, excessive daytime sleeping, depression, beta carbolines, cognitive impairment. This is the effect of this drug acting on GABA B receptor. This is the GABA B receptor. Like you can baclofenes B positive antagonist, agonist, positive modulator, antagonist. You can see this is the example of this. Baclofenes is selective GABA receptor structure analogs of GABA used as a muscle relaxant. Antagonist cyclophanes 2-OH cyclophanes CGP35 produce only minimum effect on CNS function, main effect anti-epileptic actions in animal model of absence seizure. Okay, enhance cognitive performance, therapeutic uses in human not proven as yet. Drug acting on GABA C receptor tried in RX of the residual sleep cognitive disorder. Okay, this is the selective agonist antagonist CACA cis4 aminochrotonic acid CAMP. This is type of GABA C receptor drug. Thanks. If you like this video, please subscribe my channel, hit the bell icon. If I drop any video, you can get the notification of that video. Please subscribe my channel and subscribe, likes and share. Don't forget subscribe channel. Okay, don't leave it without it. Thanks if you like. Thanks.